Hello, everyone. I'm Lydia Kerkerian-Le Goff, researcher at the Developmental Biology Institute of Marseille and member of the international group of NeuroMarseille. It's my great pleasure to welcome you to the second scientific session of this symposium, which will focus on adult neural stem cells and adult born neurons in the brain. Our speaker will share with us their recent findings on the mechanisms that govern the property and fate of these cells, and thereby the fascinating form of neuroplasticity represented by adult neurogenesis. I would like to remind you that each 20-minute talk will be followed by a 10-minute discussion, and I invite the participants to ask their question by raising their hand. They will be then allowed to activate their camera and microphone. As a first speaker, we are pleased to welcome Giuseppe Lupo, who is Associate Professor in the Department of Biology and Biotechnology Charles Darwin at the Sapienza University of Rome. He will talk us about the regulation of adult neural stem cell proliferation by an aging-related homeobox gene.
So thank you very much for this opportunity to talk about our work at this nice meeting. And today I will present recent data from our lab about the role of the homeobox gene DBX2 in the regulation of adult neural stem cell proliferation. It is well established since several decades that neurogenesis in mammals continues beyond development, also in the adult brain, but only at the level of specific neurogenic niches where special signals from the microenvironment can support neural stem cell survival, proliferation, and neuronal differentiation. One of the best characterized adult neurogenic niches is the subventricular zone, located in the walls of the lateral ventricles within the cerebral hemispheres. And here, a resident population of neural stem cells is able to proliferate, generating progeny that migrates to the olfactory bulb, where they differentiate into newborn interneurons, which integrate into pre-existing circuits and confer plasticity to these circuits and the ability to be modulated by external stimuli. Subventricular zone neural stem cells, which are represented here in red, do not generate newborn neurons directly, but through intermediate cell populations, which together comprise a cell lineage known as the neurogenic lineage. Neural stem cells lie at the top of this lineage, and they are mostly quiescent, arrested in G0, but under appropriate stimulation, they can be activated, enter the cell cycles, divide to generate transit amplifying progenitors, or TAPs, which can also divide a few times to generate neuroblasts, which then generate newborn neurons. Therefore, the adult neurogenic lineage of the subventricular zone includes cell population with different cell cycle activities. A remarkable feature of this neurogenic process in the subventricular zone is that it dramatically decreases during aging. So if we look at the subventricular zone in young adult mice, we can find plenty of TAPs and neuroblasts and a conspicuous neurogenic output. But if we look at the age subventricular zone, then we find sparse TAPs and neuroblasts and a severely reduced neurogenic output. And consequently, the olfactory functions in aged mice are compromised, which leads to the idea that the decline during aging in adult neurogenesis may be linked with the cognitive decline of elderly subjects. Different models have been proposed to explain this neurogenic aging of the subventricular zone. Initial models suggested that this neurogenic decline is due to the progressive exhaustion of the neural stem cell population. So it was thought the neural stem cells become activated at a constant rate throughout life, and this progressively consume the neurostem cell pool, resulting in reduced neurogenesis. However, recent studies suggest that even in the age niche, there is a substantial neurostem cell population, which is, however, much more difficult to activate compared to the young neurostem cell population. So more recent models suggest that increased quiescence of neurostem cells is a main driver of the neurogenic aging of the subventricular zone. To understand the molecular mechanisms that control neurostem cell quiescence and activation and their alterations during aging, recently several labs have compared the transcriptome of quiescent and activated neurostem cells derived from the young adult subventricular zone in mice, or the transcriptome of young adult and aged neurostem cells. And I'll show here a couple of examples of these recent studies. The cartoon on the left is taken from recent work by the lab of Isabel Fariñas, comparing the transcriptome of quiescent and activated neurostem cells. Red dots in this cartoon represent genes that are downregulated in quiescent neurostem cells, whereas blue dots represent genes that are upregulated in quiescent neurostem cells. And the downregulated genes, many of the downregulated genes are involved in cell cycle progression, whereas many of the upregulated genes are involved in the response to external stimuli, and in particular to inflammatory response. And interestingly, similar changes were also found when comparing young adult and aged neurostem cells. The cartoon on the right is taken from recent work by the lab of Ana Martin Villalba. And what this cartoon shows is that in aged neurostem cells of the subventricular zone, which have increased quiescence, there is an upregulation of the inflammatory transcriptome and the downregulation of the cell cycle transcriptome. And similar results have also been shown by other studies. So increased quiescence, increased expression of the inflammatory transcriptome, and decreased expression of the cell cycle transcriptome appear to be milestone of neural stem cell aging, raising in turn the question of which transcription factors could be involved in mediating these effects. We have tried to approach this question by using in vitro culture systems of mouse neural stem cells, which represent a convenient experimental system with several advantages, especially for a small lab on a limited budget, because these neural stem cell cultures can be grown using chemically defined, simple, inexpensive culture conditions, both in monolayer and as three-dimensional cultures. These cells have a high genetic stability, so they maintain a normal karyotype even after long-term passaging, but they are amenable to genetic manipulation to study gene functions. When neural stem cell cultures are grown in the presence of growth factors in non-adherent conditions, neural stem cells proliferate to produce three-dimensional aggregates that are known as neurospheres. And interestingly, neural stem cell cultures derived from the subventricular zone of aged mice grow as neurosphere less efficiently than cultures derived from young adult mice. So this in vitro system may represent a useful paradigm to investigate the intrinsic molecular changes taking place in aged neural stem cells. And indeed, we use this experimental system in collaboration with the lab of Peter Raggan at the Babraham Institute in Cambridge to compare the transcriptome of aged and young adult neural stem cell cultures derived from the mouse subventricular zone, and by means of RNA sequencing. We identify a few hundred genes that were differentially expressed and which are represented by the purple dots in this plot. And because we were mainly interested in transcriptional regulators, we specifically focused on one of these genes, which is DBX2, which codes for a homeodomain transcription factor. And this upregulated in aged neural stem cell cultures, as shown by the charts here on the left, The top chart in A shows the expression of DBX2 in vitro cultures of aged and young adult neural stem cells, whereas the chart in B shows its expression in aged and young adult neural stem cells freshly sorted from the subventricular zone and immediately analyzed without in vitro culture. And in both cases, you can see a clear increase of DBX2 expression in aged samples. The images here on the right, provided by the lab of Paola Bovolenta in Madrid, show instead the expression pattern of DBX2 in the subventricular zone by in situ hybridization, and as a positive control, also the expression in the embryonic neural tube, because DBX2, as many other homeobox genes, is also involved in neural development, but until this study, nothing was known about its role in adult neurogenesis. We have also taken a look at recent transcriptomic datasets that, as I said, have compared the transcriptome of aged or young neural stem cells purified from the subventricular zone. And in these datasets, when looking for DBX2, we have also found an upregulation in aged neural stem cells, confirming our data. But we've also looked at datasets comparing quiescent and activated neural stem cells. And interestingly, we found that the DBX2 is more expressed in quiescent neural stem cells than in activated neural stem cells. Here shown are the results with 3 different datasets from the labs of Isabel Fariñas, Fiona Dech, and Luis Parada, and they all show a convincing difference of DBX2 expression between quiescent and activated neural stem cells. So this data suggests that DBX2 might be a candidate regulator of neurogenic aging, possibly acting by facilitating the increased quiescence of aged neural stem cells. To test this hypothesis, we overexpressed this gene in cultures of young adult neural stem cells by creating transgenic cell lines and using 2 different approaches. We initially created lines with constitutive DBX2 overexpression, using as a control lines expressing GFP. But then we also generated lines with doxycycline-inducible DBX2 expression to perform acute overexpression experiments with a limited time course. And for both approaches, we produced 2 independent sets of cell lines using neural stem cell cultures derived from different animals, and repeated all the experiments using both sets of cell lines. So when DBX2 overexpressing cultures, either constitutive or inducible, are grown as neurosphere, we detect a clear growth phenotype, because the neurospheres produced by DBX2 overexpressing stem cells are smaller than control cultures. But DBX2 does not affect cell viability, so levels of cell death are similar in DBX2 overexpressing and control cultures, suggesting that this growth phenotype is mainly due to affected proliferation and not to cell death. And in particular, we thought that there might be 2 possible mechanisms of this phenotype. The DBX2 might cause cell cycle exit and arresting G0, or it may decrease the division rate of cells that stay in the cell cycle. So to analyze the effect of on the cell cycles, we use 2 different approaches. We use flow cytometry to quantify the fraction of cells in the different phases of the cell cycle, G0, G1, S, and G2M, based on DNA content. And we also perform immunofluorescence analysis with cell cycle markers, in particular Ki67, which is expressed in cells in the cell cycle, but not in cells arrested in G0, and phosphorylated histone H3, which is a marker of mitotic cells. So we first performed this analysis on cultures with constitutive DBX2 overexpression, and in this case, we did not detect an obvious effect on cell cycle exit. So the proportion of cells in the G0, G1 fraction and the proportion of Ki67 positive cells was similar in DBX2 overexpressing or GFP expressing cultures. However, surprisingly, we detected an increase in the G2M fraction, which might suggest increased mitosis of DBX2 overexpressed in culture, but actually quantification of phosphorylated histone H3 positive cells shows that mitotic cells are decreased in DBX2 overexpressed in culture. So our interpretation of this data is the DBX2 overexpression impairs the G2M transition, causing neural stem cell accumulation in G2. This data would suggest that DBX2 specifically acts on the G2M transition, but if the overexpression of this gene were promoting cell cycle exit, cells arresting in G0 would be negatively selected during the production and the amplification of cell lines with constitutive DBX2 overexpression. So we could miss a potential effect on the G1S transition with this approach. For this reason, we decided to use inducible cell lines where we could perform acute DBX2 overexpression for a few days, followed by immediate analysis of the cell cycle. And in this case, in these conditions, we did detect an effect of DBX2 in promoting cell cycle exit, as shown by the increased fraction of cells in G0, G1, and the decreased fraction of Ki67 positive cells arrested in G0. In these conditions, we again also detected a decrease in the fraction of phospho H3 positive mitotic cells. So altogether, this data suggests that DBX2 can inhibit neural stem cell proliferation by impairing both the G1S and the G2M transitions. Because it is a transcription factor, we expected that this effect were mediated by transcriptomic changes. And for this reason, we compared the transcriptome of DBX2 overexpressing and control cultures using RNA sequencing. We compared the transcriptome of cell lines with constitutive overexpression of DBX2 or GFP, and these effects are shown in the volcano plot on the left, and also the changes detected in inducible cell lines grown in the presence or in the absence of doxycycline. I would like to remind here that for each of these analyses, we didn't use a single cell line, but two sets of cell lines derived from different animals. And the red dots in these volcano plots represent differentially expressed genes. The genes on the right are upregulated genes. The genes on the left are downregulated genes. And as you can see, both in the case of constitutive or acute DBX2 overexpression, there are many differentially expressed genes. To focus on changes that were DBX2 specific and not cell line specific, we merged these two transcriptomes and selected the genes that showed the significant expression changes in both transcriptome with the same direction of fold changes. So genes that were either upregulated or downregulated, both upon constitutive and acute DBX2 overexpression. This yielded a list of approximately 1,500 genes, which we define as a core DBX2 associated transcriptional signature. And we analyzed this signature by using gene ontology databases to identify functional gene categories significantly modulated by DBX2. I won't have time to discuss now all the results of this transcriptomic analysis, but I'll be happy to provide more details after the talk. Now I would like to draw your attention on the fact that gene ontology terms enriched for genes downregulated by DBX2 include terms related to neural precursor proliferation and cell cycle progression, whereas terms enriched for genes upregulated by DBX2, sorry, I lost the pointer, include terms related to the inflammatory response. We took a closer look at the cell cycle genes affected by DBX2. This cartoon is a diagram showing the main genes involved in cell cycle regulation. The genes highlighted in green are genes downregulated by DBX2, and there are many essential components required for cell cycle progression. for example, several cyclins and cyclin-dependent kinases. Whereas genes highlighted in red are genes upregulated by DBX2, and these include a few key cell cycle inhibitors such as P21 and GAD45. Finally, in this last slide, I also show you the results of a different type of analysis which we perform, which is a gene set enrichment analysis. In this analysis, known gene sets that are involved in various biological processes and that are available in the molecular signatures database were compared with the whole transcriptome of DBX2 overexpressing cells ranked based on the fold change. So genes strongly upregulated by DBX2 are at the top of the rank, and genes strongly downregulated by DBX2 are at the bottom of the rank. And here in this bar plot are shown the top 20 gene sets showing a significant correlation with the DBX2 transcriptome. The gene sets which show a positive normalized enrichment score are gene sets that are enriched for genes upregulated by DBX2. Instead, the gene sets that have a negative enrichment score are gene sets enriched for genes downregulated by DBX2. And the gene sets with a negativeness score are mostly gene sets related to cell cycle progression. For example, targets of E2F factors are genes that are involved in the G1S transition. And as you can see, there are also gene sets involved in the G2M transition and MYC target genes. Instead, among the gene sets with the positiveness score, many of them are related to inflammatory response to pathway related to interferon and other pro-inflammatory cytokines. And there are also gene sets related to cell cycle inhibition, for example, P53 pathway genes. In conclusion, today I've shown you data showing that the DBX2 homeobox gene is upregulated in quiescent and in aged neural stem cells of the mouse subventricular zone. I've also shown you that DBX2 overexpression in young adult neural stem cell culture can inhibit their cell cycle by impairing both the G1S and the G2M transition, and that neural stem cells overexpressing DBX2 show key transcriptomic changes associated with neural stem cell quiescence and aging, and in particular, a downregulation of the cell cycle transcriptome and an upregulation of the inflammatory transcriptome. Data that I did not have time to present today suggests that the EGF receptor signaling pathway is involved in DBX2 modulation during subventricular zone aging. In particular, this pathway can repress DBX2 expression, and this pathway is involved in promoting neural stem cell activation and is known to become less functional in the aged subventricular zone. So the decreased activity of the EGF receptor pathway during aging might be responsible both for increased DBX2 expression and increased quiescence of aged neural stem cells. Finally, I would like to thank people that played a fundamental role in this project. Andrea Giuliani and Paola Nisi are former students in my lab who did most of the work with transgenic cell lines. Valerio Licursi gave an outstanding contribution by doing all the bioinformatic analysis. Mario Fiore gave essential help with flow cytometry analysis. Emanuele Cacci runs his lab next door to mine, and we collaborate on many projects, including this one. And as I said, the initial screening leading to the identification of DBX2 was performed in collaboration with the lab of Peter Raggan at the Bagram Institute in Cambridge. Thank you very much for listening, and I'll be happy to take questions.
Thank you very much, Giuseppe, for this very interesting talk. So there is a question from Olga Garaschuk.
Hi, Giuseppe. Beautiful talk. Thank you so much indeed. My question was, what is the cause, what is the consequence? Because aging organism upregulates probably everywhere, not only in the subventricular zone, all these gene pathways connected to inflammation, inflammaging, and whatever you have on your list. So how is your gene getting into this game? Is this a cause, is this a consequence, or where to put it?
Thank you very much for this question. It's a very important, very interesting question. So it is known that in the aged subventricular zone, there is increased inflammation because there are systemic pro-inflammatory cytokines that get there and can activate an inflammatory response in stem cells. So we thought of a simple model where increased levels of these cytokines can promote DBX2 expression, and this factor could act as a mediator of these cytokines. But actually, in vitro, we find no effect of several cytokines that we have tried on DBX2 expression. However, there are papers showing that when you induce quiescence in neural stem cells in vitro, for example, work from the lab of Steve Pollard, in the absence of any pro-inflammatory cytokine, you can upregulate the inflammatory transcriptome. So there is the idea that these genes might have a role in the regulation of neural stem cell activation or quiescence that is independent from their role in inflammation. And this is a possibility that will have to be explored with additional work.
Thank you. There is a question from Pascal Durbeck.
Thank you for this really nice presentation. I was wondering, since TBX2 through activation of the FGF pathway has been shown to be important for cell fate acquisition during development, if you have tested the cell fate of your neurosphere or neural stem cells culture after TBX2 overexpression, do you change the ratio of neurons, oligo, and astrocytes?
Yes, thank you. This is also a very important question, which was also asked by the reviewers of the first manuscript that we published 3 years ago. And actually, in the presence of growth factors, we don't see any differentiation either in the presence or in the absence of DBX2. So it doesn't seem to be an effect caused by the induction or the modulation of differentiation, but a specific effect on the cell cycle of cells that retain stem cell identity. In the future, it will be very interesting to study the effect of DBX2 also on the ability of stem cells to differentiate along specific lineages.
Okay, thank you.
Giuseppe, there is a question in the Q&A.
Sorry, there is a question.
In the Q&A.
Ah, okay, yes. Okay. Okay, so the question is asking whether in the human subventricular zone, cells contribute to olfactory neurogenesis, or instead they move to the prefrontal cortex. And in the same question also, it is asked whether subventricular zone stem cells might participate in the repair of the brain following injury. Okay, so I'm not directly involved in research into these aspects, but as far as I know, in the case of the human subventricular zone, there is a sharp decline in neurogenesis after infancy. So in adolescence or older individual, it has not been possible to find evidences of active neurogenesis. This, of course, does not rule out the possibility that you have quiescent neural stem cells that might be reactivated in some way to contribute to, for example, therapeutic effects. And then, yes, it is true, as far as I know, that the subventricular zone in mice, when there is a brain damage, for example, after stroke, can produce cells that can migrate to the site of the lesion, but as far as I know, they don't seem to contribute to repair or regeneration. So, but it might be possible to find the right way to stimulate these cells to generate neurons and integrate into the damaged circuits after damage, since they reach the damaged area. Thank you.
You have a question from Santiago.
Yeah, thank you very much, Giuseppe, for your nice talk. Very quick question. Do you have any evidence that TBX2 may affect telomerase activity and the senescence of NPCs?
This is a very, very nice question. No, we don't have evidence about that. We haven't analyzed this aspect. We would love to do that. We are not experts on telomeres, so we would love to do it in collaboration with an expert on this aspect.
Well, there are quite a few in Marseille, I guess.
Thank you.
Thank you.
I have also a question, Giuseppe. Do you think that the reverse experiment would give rise to the reverse result? So down-regulating this factor in aged cell could reverse the phenotype?
Thank you very much for these questions. This is an experiment that we have tried to do using a knockdown approach, so short hairpin RNAs. Unfortunately, although we tried several sequences, we couldn't find anyone that efficiently knocked down DBX2 transcripts. So we get a decrease of at most 20 to 30%, which is less than the change that we see between aged and young cells. And in this case, we did detect opposite changes in the expression of several genes, but there were also many other genes that were not affected, and there was no proliferation phenotype. So we will need to do a more effective knockout or dominant negative approach. But also I should say that when neural stem cell cultures are grown with growth factors, they proliferate very efficiently. So it might be difficult to detect an effect that causes increase of proliferation. So we might also need to devise different types of experiments, for example, induce quiescent and knock out DBX2 and then see whether there is a change in the quiescent state of the cells rather than just looking at proliferation in the presence of growth factors. But really good question. Thank you.
Thank you, Giuseppe. So we have to close this discussion. I would like to encourage also the participants to raise a question, to address a question by raising their hands. So not only the panelists, but everyone in the audience can raise their hand to ask their questions. So we will now move to the next presentation, which will be delivered by Professor Dr. Olga Garaschuk, who is head of the Department of Neurophysiology at the University of Tübingen. Our talk is entitled Molecular Determinants of Sex Selection in Adult-Born Neurons.
Okay, thank you so much indeed. Then I can proceed. So I am so thankful for Giuseppe for introducing the whole circuitry here. Therefore, I will not be going from the subventricular zone. I will just introduce the rest of the circuitry from the other more sensory part. So we are looking here at the sensory neurons in olfaction, and we are staying in the olfactory neurogenesis system. And we see here that those cells project to the olfactory bulb. And the first synapse from the sensory neuron to the output neuron of the olfactory bulb, so-called mitral or tufted cells, is in this structure named glomerulus. And because we are talking about neurogenesis today, we will be talking about two different types of adult-born neurons. One type are granular cells, and the other type is even not drawn on this scheme. These are juxtaglomerular neurons. However, juxtaglomerular neurons are the third large population in our brain after the granular cells of the cerebellum, granular cells of the olfactory bulb, and then juxtaglomerular neurons are coming into the game with approximately 1 million cells in the olfactory bulb. And those cells basically are part of this neurogenic niche, which starts here in the subventricular zone, and thereafter neuroblasts are migrating along the rostral migratory stream, and then they are entering olfactory bulb. And here, some 90% of those cells become olfactory bulb granular cells, and some 5 to 10% become juxtaglomerular neurons. Because juxtaglomerular neurons are located on the surface of the bulb, it's very easy to study them in vivo, and therefore they will be our major target for analysis today. And in general, adult-born neurons in the olfactory bulb are critically important for associate learning of difficult tasks, fine odor discrimination, olfactory memory, and fear conditioning, just to name a few the tasks where those cells are important. There are more. And today, for this short talk, I would be focusing on mechanisms of controlling migration, differentiation, integration, and survival of the cells under in vivo settings. So what we will be doing, we will be labeling those cells as they are migrating in the rostral migratory stream, either with retroviruses encoding different combination of viruses, and I can show you already how it looks like. So this has 3 different color, red, green, and blue cells. And if you put viruses encoding all these 3 colors together, then you can label stochastically different cells in different colors, which is actually very nice because it gives you basically unique ID for every cell. And because cells are migrating, you can follow their migration and learn a lot about their behavior. Or alternatively, we will be labeling those cells with the same approach in the rostral migratory stream with a calcium-sensitive calcium indicator to measure their activity. And when doing this study, which was published in Cell Research already in 2016, we learned basically some general principles of how those cells are migrating and what they are doing during their so-called pre-integration phase. And pre-integration phase is basically the phase when the cells are already arriving at their layer of destination, glomerular layer, if these are juxtaglomerular neurons, or granular cell layer, if these are granular cells. And then the question is, what are they doing between the arrival and their final settling in this network? And this is basically the pre-integration phase. And what we learned is that basically after tangential migration in RMS, both cell types, granular cells and juxtaglomerular neurons, are entering radial migration, but here their ways basically differ. While granular cells just move up and stop in that position in the granular cell layer, juxtaglomerular neurons basically shoot up, speed up here, because here probably the decision is taken, am I a granular cell or do I want to become a juxtaglomerular neuron? And if they really decide to become a juxtaglomerular neuron, they move very quickly through the external plexiform layer, and then they are entering basically 2-week-long journey inside the glomerular layer. And this journey is extremely complex, so cells may move up to 1 millimeter if you just add together all this pathway, and this is so-called lateral migration. And this lateral migration can be very complex. It can be unidirectional, like in this case, right, going straight to its destination. It can be multi-directional, kind of moving around and wondering where should I integrate. But altogether, it takes up to 2 weeks. So cells after their birth, they are approximately at day 7 or 8 at the glomerular layer, and then around day 20 they are probably settled. Up to 30 days, 30 after birth, they are for sure settled in the right place in the glomerulus. And however, while migrating in this basically juxtaglomerular cells, while migrating, they do already functionally integrate in this network. And this functional integration is extremely fast, as we could show in this study here. This is one cell which we monitor in live. Cell is migrating in the glomerular layer, and we are stimulating nearby cells with odorant, and we see this cell is not responding. But only 2 hours later, the guy starts it gains this response to an odorant. So from one hand side, the guy is wondering for 2 weeks where to integrate. On the other hand side, when it decides when and where to integrate, it does it very quickly. So to study the mechanisms of this migration and differentiation and integration, we have developed a system where we basically can modify the intrinsic activity, either overexpressing the potassium channel KV1.2, voltage-gated potassium channel, or overexpressing the inward rectifier KIR2.1 channel. So this experiment is supposed to regulate the pattern of cell firing. This experiment is supposed to check for sustained activity level, background activity level of the cells. And then as a control, we are using basically the same calcium indicator, which will be our functional readout, but then put nuclear cherry just to keep the gene load the same. And just quickly going back, what is really very unique to these juxtaglomerular neurons, that all other cells, be it in development or granular cells in the olfactory bulb, which are adult born, they all first migrate and then start growing their dendritic tree. And this is very different for juxtaglomerular neurons. They basically doing both processes together. And you can appreciate here in the very same experiment described here, we can see just in 4 hours how dramatically the shape, the morphology of this cell has changed. Okay, so the first question for us after doing this modification was, so what is an impact on migration? If we are modifying intrinsic activity of adult-born neurons, how does this impact their migration? And this is some representative example here for control cells labeled with this construct and for KV overexpressing cells, and you can see already appreciate that it seems like something is different. To quantify this, we went through very detailed analysis. And first of all, if we look at migration of adult-born neurons, this migration is saltatory. So here we can see this because each dot is basically location of the cell in line. So this is a cell, this is 4 hours, and this is the speed. So we see the high speed of migration, then stops migrating, and then continues, and so on and so forth. So we have saltatory migration, and then we have analyzed lots of different parameters, be it fraction of migrating cells, be it maximum migrating speed, medial migrating speed, translocation. All those parameters were dramatically down-regulated by modifying or basically inhibiting intrinsic activity of these neurons. So everything is going down. So cells are migrating less easy, so migration is impaired. And because maybe not every cell was migrating during 4 hours when we were recording, then we also analyzed the fraction of migrating cells between day post-injection 8 and day post-injection 11 in 3 days, just to give more cells chance to contribute to this migration. But also this kind of migration is pretty much impaired. And then we have asked, okay, activity is important and activity is important for migration. Is this just endogenous activity, or maybe any activity can do it? An alternative way to ask this question will be the odor deprivation. So basically asking whether sensory driven activity has the same impact as the endogenous activity. And if one looks through the literature, actually the hypothesis will be that sensory experience is much more important than endogenous activity for these cells. And we remember cells acquire this sensory responsiveness very quickly. So to do these experiments, we closed the nostril of the animal with this plug. We did it at day post-injection 5, so even before cells arrived to their destination level. And we kept it till day post-injection 20. And then we checked at day post-injection 20, and those animals which were analyzed were supposed not to have responses to odorants through this closed nostril, even at day post-injection 20. And then if we remove such bulbs, we can see, and this is staining for tyrosine hydroxylase, and tyrosine hydroxylase is a nice activity marker, so for the whole bulb, and we see that the tyrosine hydroxylase level is decreased in odor-deprived hemibulb, and actually, thanks to anatomy, we can take one animal for control and experiment, because air pathways are pretty independent to the left and to the right bulb. So we have an intrinsic control, so to say, for everything else which might happen, but this nostril closure, this odor deprivation. And then we basically did very same analysis I've shown you in the first slide, looking at fractional migrating cells, the median speed, maximal speed, translocation, and fractional migrating cells over 3 days. And none of these parameters was changed under sensory deprivation, telling us that this is endogenous but not sensory-driven activity, which is important. Next, we asked about the morphology of those cells, and this morphology we have analyzed a day post-injection 20, when the control cells have already very elaborated morphology. And this was very strikingly different in our two groups with KV and Cure group, because their cells could barely show any processes, any dendrites. And as expected, if you look at these pictures, the total dendritic branch length and number of branches and all this shoal analysis showing us the complexity of the cells basically were pretty much downregulated in KIR and KV groups, telling us that modifying or inhibiting intrinsic activity also impairs dendritogenesis or morphogenesis of those cells. This was not the case for odor-deprived animals. Here we couldn't find any difference between contralateral part and occluded odor-deprived ipsilateral side. Then we asked about the cell death, basically, and here for my friend Harold, we did it very accurately. So we were monitoring, and this is our basically experimental timeline. We started retroviral injection at day post-injection 0, and then we were handling and training animals just to record everything in non-anesthetized animals, and we were recording those cells, and these are images of those cells labeled with RGB viruses, and then we were doing basically many sessions of 2-photon imaging in vivo. And then basically our criteria, we counted as dead cells only those cells which either were really seen that they leave some cell debris, and if we had this multicolor labeling, then usually M-cherry, because it's processed less slowly than other proteins, it turns over slower than other proteins. Usually when cell was dead, we still could see some remaining of M-cherry, but not of other proteins. And then basically either we really see the debris of this cell, or in older animals after DP20, we also accepted another criterion. Basically, if cell already settled and was stable for more than 4 days, and the day post-injection was more than 20, then after sitting so long in the same place, if the cell is disappearing, it also was counted as dead. So very restrictive criteria to take care for eventually cells moving out of the field of view. We also were only analyzing cells in the center of the field of view, never touching cells basically at the edges, and calculated the possible migration speed per day, looking at our migration data. With all this care taken, we can provide the following numbers: that surviving cells during this time period, and this is a critical period for cell integration, so-called pre-integration phase. For this period of time, 60% of our adult-born neurons survived, but rather around 20% of our cells were eliminated, and these were really very, very restrictive criteria. So likely that this percentage is higher, but then this we cannot say for sure. And we published this data and also lots of other data about factors predicting elimination of adult-born neurons in this manuscript. I don't have any time to talk about it today, but please look it up if you are interested. And I am continuing with basically with integration of our adult born neurons with modified or inhibited intrinsic activity. And what we can see here, we just splitted it into two parts: survival, which is more difficult to measure during this early phase when cells are migrating, and then survival over the later phase when cells really have already settled and after that were dead. And in both cases, we see that survival is less in KV 1.2 and KIR 2.1 groups, and there is a significant difference for both groups to control group in this case, and there is a significant difference only in this case. So it looks like later only cells in this KIR 2.1 group are surviving worse than control cells. Okay, and next story, what about the functional integrating into pre-existing neural network? And in terms of functional integration, we looked at their odorant responses and basically, first of all, analyzed the fraction of odor-responsive cells, and this fraction is decreased significantly in both groups, and also analyze the amplitude and area under the curve of this odor response, and also both these parameters are reduced. And then after having all this data, we wondered, okay, so intrinsic endogenous activity is so important. We know we inhibit the endogenous activity, but how did we change it? What is really different? And then we started analyzing the curves of this activity, basically recorded here for 15 minutes in different cells, in control group, KV group, and KIR group, and also in this contralateral control group from our odor-deprived mice and in the ipsilateral odor-deprived group. And what we can see already from these curves that basically what is happening, that there are lots of fluctuations in different cells in terms of calcium signaling, which are preserved under odor deprivation in control groups, but are basically pretty much gone in both KV 1.2 and KIR 2.1 group. And we have analyzed this in much detail with mathematical approaches, but basically the message is as clear as it is here. So we are indeed losing in these two groups calcium fluctuations. And this is not the case for odor deprivation, consistently to our previous results. Okay, so what to do now? Now we were interested in mechanisms underlying these findings, and there we went for the RNA-seq data. So basically we labeled those cells, they were already labeled with Twitch2B, and this is a green fluorescence, and we sorted by this green fluorescence, and then we analyzed the cells with RNA sequencing, and what turned out that basically we had lots of pathways important for PCREB-driven gene expression, for example, calcium calmodulin pathway, PCREB-driven expression of immediate early genes, but also some GOs, which are gene ontology pathways, which are important for neuronal migration, neuronal differentiation, dendritic morphogenesis, all were decreased or downregulated in these two groups, KV and KIR groups, but the pathway for apoptosis was upregulated and internal controls were unchanged. With all data integrated, we were coming to this scheme, basically, starting with voltage-gated calcium channels, which were downregulated. Also, NMDA receptors can be placed here. Also, the expression of those was downregulated. And then via this pathway, basically transmitting this decreased activity on the cell membrane to the nucleus via chemkinase 2, calcium calmodulin pathway, and chemkinase 4, basically changing or phosphorylating CREB to become phosphorylated CREB. And then also it was an interesting pathway of this CREB phosphorylate transcriptional co-activator, CRTC1, which is basically making a complex with PCREB to help basically to start expression, for example, of immediate early genes or other PCREB activated genes. So all these players were downregulated, while the genes encoding for apoptotic pathways were upregulated, and control genes were basically housekeeping genes were not changed, and these stars mean that these data were validated by PCR. Okay, so just to validate this finding, which was a sequencing mRNA-seq data, we went to look for pCREB, and indeed, staining for pCREB, we could show that basically in adult-born neurons belonging to KV1.2 and KIR2.1 groups, the CREB phosphorylation is dramatically downregulated. This is not the case for these two groups, and it happens if analyzed at DPA10 when CREB phosphorylation is very high, but also when looking at DPI28 when CREB phosphorylation in a usual control cells is going down. We can see here these levels here, fluorescence. Okay, so let's summarize. What did we learn from this story? We learned that migration, morphogenesis, survival, and functional integration of adult-born juxtaglomerular neurons during the pre-integration phase are activity dependent, are primarily driven by patent endogenous but not sensory driven activity, rely on activation of voltage-gated calcium channels and also NMDA receptors. We have this data in the paper. You can look it up on bioRxiv. They need fluctuations in intracellular calcium concentration, which is driving activation of CAMK2 and calcineurin-related pathways and causing pCREB-mediated gene expression. This need for calcium fluctuations is likely imparted by salt-inducible kinase 1, SYK1, which phosphorylates CRTC1. So CAN is actually dephosphorylating it and bringing it to the nucleus to start gene expression, and SYK1 is phosphorylating it and triggering its export from the nucleus, and in this way arresting the pCREB CRTC1 mediated gene expression. And then there is an interesting twist to this story, that basically what we are doing here to block endogenous activity was kind of making, basically up-regulating the function of KV1.2 and KIR2.1, so basically mimicking their gain of function. And interestingly, such gain of function mutations in both basic KV1.2 and KIR2.1 channels are indeed known from the neurology, and they are causing developmental epileptic encephalopathies. And interestingly, for example, for KV1.2, the loss of function, because we are thinking that this channel is important for repolarization of cell membrane, the loss of function is causing much less symptoms than gain of function. And if one comes from the idea of the network, which is hyperactive during epilepsy, then it seems not natural that driving this network down makes more epilepsy. But if we are coming from our findings and thinking about this impaired migration, morphogenesis, and functional integration of, in our case, inhibitory neurons, because I forgot to mention that all cells we were looking at, adult-born juxtaglomerular neurons, are to 95% inhibitory, then we might have very different, basically, network to play with or framework to think about those upregulations, those gain of function of those channels, and their role in developmental epileptic encephalopathies. So with this, I am done. Thank you so much for your attention, and I'm happy to take your questions.
Thank you, Olga, for this great talk. So the talk is open for discussion. Pascal?
Thank you, Olga, for this really great presentation. I have a question. When you are modify migration of this neuroblast modify intrinsic activity, so these cells which have been modified, and you observe a modification during migration, do you think that this the mode of migration is also modified? So in other words, do you know if these cells are still in chains? Are they still embedded in the astroglial network?
So what you are telling, thanks for this very interesting question. So what you are thinking, it's migration in the rostral migratory stream, yeah? So tangential migration. We haven't looked at that. It's difficult to do in vivo simply because it's deep, but we would not expect much modification there, and also during the radial migration up, because we counted what we did to answer a similar question. We counted the rate of arrival of those cells to the glomerular layer, and this rate of arrival is similar for all three groups. This data are on the paper is on bioRxiv, so you can look it up. So and this is basically also clear because it seems like voltage-gated potassium channels are not expressed yet in this early period. So what we are modifying with our tools is just this lateral migration during the pre-integration phase, when cells are already at their destination layer but have not integrated yet in a place where they are due to integrate.
Okay, thank you.
Olga, do you think that default in morphogenesis are linked to a mispositioning of the cells?
So we can't answer this question because we happen to modify with one single change. We happen to modify all of that, yeah? Okay. So, but we can't, therefore we can't differentiate. We don't know whether we can modify them differentially. Yes.
Santiago?
Yeah, very nice talk. Thank you, Olga. I was wondering, since you've been working also pretty much with Alzheimer's models, whether some of the integration deficits problems that you found in this study could be applied somewhere to the deficits in olfaction that we observe in men, but also in animal models.
Theoretically, yes. Practically, we are studying, as you say, because we have this fortunate combination of competency and expertise in our lab. We are studying exactly the question you are asking. It looks like this, in mouse models of Alzheimer's disease, this impairment of olfaction, which we know from behavioral studies, has its cellular molecular basis already very early in the game. And at least those ones we have looked at, there are additional ones to adult neurogenesis, let's put it this way, because we haven't made the very same study in mouse model of Alzheimer's disease, but we have done some other studies there, some functional studies, and we found additional mechanisms. But I could imagine that also we know that adult neurogenesis is decreased, and what is the mechanistic reason for this decrease? This we don't know yet.
Okay, thank you.
Thank you.
Are there any questions in the audience? Do not hesitate to raise your hand.
There is one in the
Q&A.
Yes. Could I precise the role of Syk in the migration of cells? This is the role of Syk is not for migration. It's more like what we basically seem to have learned. We seem to have learned that calcium fluctuations are key for all processes I was talking about. And this, what we have done with our study, with our intervention, we have just removed those calcium fluctuations. And then we were asking ourselves, why is it that calcium fluctuation is so important? Not the level of calcium, and there are some data from the olfactory deprivation. So there we are reducing the basal level of calcium, not significantly, but significantly, not much, but significantly. But this doesn't have any importance. So at the very end, what seems to be very important is a fluctuation. And then how can we explain that calcium fluctuation is driving gene expression differentially? And from toolkits which popped up out of our RNA-seq studies, we could imagine a mechanism where we need basically the phosphorylation of this coactivator of CREB, dephosphorylation of this coactivator of CREP, which is done by calcineurin in the cytosol, and then phosphorylation of it to drive it out of the nucleus, which is done by this SIC1. And SIC1 is basically one of genes which is expressed in a PCREP dependent manner. So PCREP starts, SIC1 is expressing, and then SIC1 is stopping PCREP dependent gene expression, further gene expression. And in this way, the calcium signaling pattern which is effective would be going up and going down, but not the one coming up and staying. So this is the logic behind. And if it was too complex now, I'm sorry for that, but it's also in this paper, so you can read it up there. We try to explain it in more words and more detail.
Thank you again, Olga. So we are now moving to our last speaker of the session with Dr. Harold Cremer, head of the team Molecular Control of Neurogenesis in the Developmental Biology Institute of Marseille. So he will talk us about the transcriptional and post-transcriptional control of neurogenesis.
Okay, thanks again, Olga, for this nice talk. Interesting and somehow contradicting results. Not absolutely. Okay, so what I will do today is talk about, yes, here we go, back and forth. Well, I'm actually not talking about adult neurogenesis in its proper term, but I'm talking about postnatal neurogenesis. That is the peak point when most of the interneurons of the olfactory bulb are made in the subventricular zone, ventricular zone, subventricular zone of the forebrain. So this neurogenic system, as we call it, contains or goes through all basic phases that a neuron has to go through to become a neuron, from quiescent radioglia that gets activated, passing through intermediate stages and amplification stages, migration and integration in the system, integration in the system. So the best way to look at this system is, to our eyes, to use fluorescent label. So we developed a method in vivo electroporation of this postnatal system. It works best almost only in postnatal animals, where we can inject DNA solutions into the lateral ventricle, as it is often done in the embryo, in the cortex. And then we apply an electric pulse and label stem cells bordering the ventricle of these animals, the lateral ventricle. So that is what we see in high resolution. So we target neural stem cells here in the wall of the lateral ventricle around birth, just after birth. Actually, these animals, we put them on ice to anesthetize them, and we almost never lose any of these animals. So what we target is radioglia stem cells, which are still present at that age. One day after electroporation, these cells start to go into proliferation. We see first cells appearing or leaving the ventricular zone into the subventricular zone. After 2 days, we have massive amplification of these cells in the so-called C cell stage. Then these cells enter the rostral migratory stream and start their chain migration into the olfactory bulb, here into the olfactory bulb. Once they arrive in the olfactory bulb, they detach from this structure, which is shown in this little movie. They detach from the RNS. They migrate radially. After this tangential migration, they migrate radially into the different target layers and become, as Olga already said, 10% periglomerular or juxtaglomerular neurons, and about 90%, the majority, becomes granule neurons in the center of the structure. And we can, with very high resolution, and Olga already showed that, follow the morphology and development of these neurons. So over the last, well, almost 2 decades, we used this, sometimes I say, as our Drosophila, as a simple model, as an accessible model to study how a neuron is generated from a stem cell to a functional neuron connected to its neighbors. We studied how neural stem cells are molecularly determined, how the correct number of neurons is produced, for example, by an interaction between long non-coding RNAs and microRNAs. how long distance migration is regulated, our entry point. NCAM was a key regulator of this process, or is a key regulator of this process. We studied how these cells leave the RMS under the influence of reelin signaling, for example, how these neurons are integrated in the system, and how they make synapses, for example, by the use of agrin, a synaptogenic protein. And we asked how homeostasis and versus growth behaves in the system, and the role in olfaction. So today I will focus on only one aspect of the whole system, which is the question of how these neural stem cells are molecularly determined. So actually this system generates, as I already mentioned, and Olga already mentioned too, different types of neurons. So different stem cells here in the ventricular, subventricular zone go into proliferation and generate different types of neurons. Some of them, many of them generate these granule neurons with this typical morphology. About 10% go to the periphery and integrate as periglomerular neurons. So basically the entire ventricle is a mosaic of stem cells. Excuse me. These stem cells are lining the walls of the ventricles, and different regions of this ventricular wall generate different types of interneurons that after the migration generate or integrate into different layers of the structure, granule cells or the glomerular layer. And there's a link between the birthplace of these cells, here for example superficial cells in this aspect versus deep cells in this aspect of the ventricle, generate different types of neurons that all make it into the structure. And for some of these neurons, we have good molecular marker, calretinine, for example. TH labeled dopaminergic neurons, calretinine labels also some granule neurons, and we have calbindin producing neurons in the periglomerular layer. So now this model is very convenient because we have access to different stem cell lineages, pools of stem cells that generate different types of neurons. And this is shown here, again, using our electroporation approach. When we label dorsal cells, so we hold the electroporation pipette like this, we label this dorsal stem cell pool, which after the migration 14 days later become superficial cells. They go to the periphery, mainly, or actually almost exclusively, and become these periglomerular or juxtaglomerular neurons. 15% of these cells express DH, dopaminergic neurons. About 18% when we label the stem cell pool express calretinine. We never see calbindin positive cells. 2% become glutamatergic neurons, and 65% become GABAergic juxtaglomerular neurons for which we don't have markers. If you do the same thing in the lateral wall here, we generate mainly deep cells, granule cells of this type. We also generate some dopaminergic neurons, about 5%, which go to the periphery. None of these cells express calretinine. About 10% express calbindin, go to the glomerular layer, and about 85% are other GABAergic neurons and essentially granule cells, the granule cell layer. So some time ago, Christophe Becklin decided to try to use this approach to get insight into the relation or the expression of gene expression into these two lineages. So what he did was he electroporated dorsal or lateral stem cells, and then after different time points after electroporation, which is our point 0, 8 hours, 1 day, 2 days, 4 days, 7 days, 14 days, he isolated these cell types, the different cell types, radial glia at the beginning, mainly type 2 cells at 2 days, migrating cells at 4 days, mainly, well, this is radial migrating cells in the olfactory bud at 7 days, and then at 14 days, young neurons from the olfactory bud, which actually becomes more difficult the older these cells become. So this is one of the results of this screen. We have dorsal and lateral lineages, gene expression for a variety of different, I have to get rid of this, yes. dorsal and lateral gene expression over time from stem cells to young neurons. We find typical cortex-related genes like PAX6, EMX, TBR1, etc. in the dorsal lineage. We find typically subpallial genes like DLX, etc., FOXP1, MASH1 in the lateral lineage from stem cells to neurons. Okay, so one of these genes attracted our attention. Yes, up here we go. This is PAX6. PAX6 is a well-characterized transcription factor, and it's expressed slightly, but predominantly in the dorsal lineage. Okay, so PAX6 is now an interesting factor because PAX6 has been implicated in the generation of dopaminergic neurons in the olfactory bulb. So this is data from Covey and Alvarez-Guila, for example, that used, so PAX6 deficient mice die at birth. At that time, conditional mutants were not available. So what they did is use bred an actin GFP allele into a PAX6 heterozygous background, and then generated wild type heterozygous or mutant mice, and transplanted subventricular zone from this tissue, from these animals that die at birth, so you can have access to this tissue, into the subventricular zone of wild type animals, and then looked at the behavior of these cells, actually at their capacity to generate dopaminergic neurons in the olfactory bulb, finding that only wild type cells are efficiently able to generate dopaminergic neurons in the olfactory bulb. Mutant neurons are not, and even heterozygous cannot do so. So it meaning that you need the right dose of PAX6, a sufficient dose to generate dopaminergic neurons. And this is a result, so it's necessary to generate dopaminergic neurons. This is a result from our group, from Nathalie Corré and Antoine de Chevigny, showing that, so when you electroporate the dorsal wall just with GFP, you get a certain amount of dopaminergic neurons in the olfactory bulb. 14 days later, you do it in the lateral wall, you get a lower amount. But if you overexpress PAX6 in the lateral wall, you increase the amount of dopaminergic neurons in the olfactory bulb, showing that overexpression of PAX6 or expression of PAX6 is sufficient to induce this phenotypic trait. Okay, so now we ended up with, we started with an interesting observation. And this is that actually the expression of the PAX6 promoter, or the presence of its mRNA, is widely distributed along the dorsal and lateral aspect. Basically, that's what it looks like, that it goes deep, the expression of mRNA, and here it's actually revealed by the human PAX6 promoter, a transgenic mouse line, goes deep into the ventral aspect. In contrast, PAX6 antibody stains only cells in a very dorsal aspect. This is somehow quantified here, that almost 80% of all PAX6 transcript positive cells in this area are positive for protein. In this area, it's about 40%. And the deeper you go at the endpoint, you don't find any, you still find mRNA positive cells, but they never express any PAX6 protein. So this looks, the protein expression is restricted to the dorsal aspect of, excuse me, of the subventricular zone. So that's a striking discovery, pointing to post-translational regulation. So we ask, where does this regulation come from? Often post-translational regulation is contained in the UTRs of an mRNA. So here we compared the expression of PAX6 only as an open reading frame to the expression of PAX6 containing its 5' and 3' UTRs, electroporated in the lateral wall of the ventricle. And then we ask, how is the expression, the relative expression of these two vectors to each other? So we see that this PAX6 open reading frame is stable over, very stable over the dorsal ventral axis. In contrast, when the same coding region is accompanied by its UTRs, it's rapidly downregulated the deeper we go along the ventral axis. Okay, so a second experiment. Here we use a MICTAC, a totally unrelated protein, compared to the same unrelated protein, but linked to the 3' UTR of PAX6. In the dorsal aspect, these two transcripts show no difference. They are both stably expressed. When we put them in the lateral aspect, we see that the MICTAC alone is strongly expressed, but the one carrying the PAX6 UTR is rapidly downregulated in the lateral wall. So there is something in this 3' UTR of PAX6 which is responsible for the restriction of protein expression to the dorsal aspect and to the downregulation of mRNA activity in the ventral aspect. So post-transcriptional regulation, nowadays we immediately think of microRNAs. So microRNAs, well, don't need a long introduction now. They're generated in the nucleus. They are modified by a series of proteins. Dicer at the end generates the active part of the microRNA, and then one of the two strands of this 222 nucleotide RNA is integrated into the so-called RISC complex, where it leads to binds to mRNA, to target mRNAs, leading to translational repression or mRNA destabilization, in any case, to absence or downregulation of protein expression. So the 3' UTR of PAX6 contains 5 microRNA binding sites. Well, listed here. And we try to find out how these are correlated with the microRNA that are expressed in the subventricular zone. So we isolated dorsal and lateral subventricular zone tissue and performed deep sequencing to see which microRNAs are expressed in these two compartments. Finding that actually only one, miR7A, is expressed at considerable levels in the lateral pool, so in this stem cell pool. To further look into depths, we performed an electroporation experiment, so labeling the dorsal and the lateral wall. So here really looking at, so this is bunch tissue, dissected tissue, so crude tissue. Here we look into the neurogenic lineage. In the dorsal lineage, we see low amounts of miR7. In the lateral lineage, we see high amounts of miR7. And actually, this is a gradient because if you look into the dorsal, medial, and ventral aspects, you see that the deeper you go, the higher the miR7 levels go. So this leads to such an idea and model that we have two overlapping gradients, PAX6 mRNA versus microRNA inhibiting this PAX6 translation and leading to protein expression only in this very dorsal aspect of the subventricular zone. So of course we need to test this. This is the first set of experiments. Actually, we performed a whole series of experiments. Here this is a part that I already showed you. So we express a MICTAC in the lateral wall, and it's well expressed in this compartment. When we hook it, the PAX6 3' UTR to this MICTAC, it gets efficiently downregulated in the lateral wall. But when we put only 2 point mutations into the miR7A binding site, specifically into this site, we recover this downregulation in the lateral wall. The second experiment is a knockdown experiment. Here we use a miR7 sponge, so a strong promoter driving a concatemer of miR7A binding sites, electroporated in the lateral wall of the ventricle. 2 days later, we see, excuse me, we see when we perform in a controlled situation that in these medium aspects, PAX6 is almost entirely absent as the protein. We see when we use the miR7A sponge, a slight upregulation, but it's significant. And importantly, 15 days later, we see an increase in dopaminergic neurons that are generated from this lateral stem cell. So this leads to this model. Actually, it's more than a model. It's how it works. That miR7 is a negative regulator of PAX6 mRNA expression, and this confines PAX6 expression to the dorsal aspect. Okay, so this was about PAX6, but of course, other cell types are generated along the lateral wall. The majority are granule cells, but there's also a population of calbindin expressing interneurons that Rebecca Erie and Alvarez Bulla have shown to be under the control of sonic hedgehog signaling. Okay, so but we went back to our screen to get an unbiased idea about the expression of transcription factors in the system, using again our dorsal versus lateral gene expression screen and looking about the expression of factors with specific expression over time and in space. One factor that pops up was VAX1. So VAX1 is a homeodomain transcription factor. It's expressed in a variety of systems, in the ventral brain widely expressed, important for eye development, etc. So Nathalie Corré looked closely into the expression of VAX1 based on this first gene expression screen, here comparing VAX1 to PAX6 expression, PAX6 in stem cells. And she always finds that there's a total exclusion of PAX6 expression and VAX1 expression. Strongly VAX1 expressing cells, so here at the transcript level, never show any PAX6 expression. On the other hand, a small pool of ASCL1, so MASH1 positive transit amplifying cells shows expression of VAX1, and most of the DLX producing migrating precursors show VAX1 expression in the subventricular zone, leading to such a pattern that in time we get a rapid upregulation of VAX1 in early stages of the proliferation process. And in space, it means that we have actually we confirmed that. So this looks like a gradient at the in situ level. You confirm this expression as a gradient by using dorsal, medial, and ventral tissue and asking for VAX1 expression. And VAX1 is expressed in a ventral dorsal gradient with highest expression levels at the lower edge of the ventricle. So, okay, is there a role of VAX1 in neuronal subtype generation? Here we collaborated with Patricia Mellon, who generated a VAX1 conditional knockout mouse. We generated a double transgenic mouse using the AF14 allele that allows labeling of cells with T tomato. And on the other hand, deletion of 2 key exons in VAX1 by Cree recombination. We used electroporation approach to be specific to our neuronal lineage. Here we had to use Cree mRNA because mRNA is, electroporation with mRNA is much more efficient, we showed that before, than expression of plasmids, Cree expression plasmids. And we needed to target this very, very, very ventral region where most of these VAX neurons are positioned. So we electroporated Cree mRNA in the lateral wall and analyzed the phenotypic outcome of this manipulation. Knockdown of VAX1 has no impact on granule cells. Once the majority, the main population is generated in the lateral wall, it has a strong impact on this calbindin producing population which migrates to the glomerular layer. So VAX1 is necessary for the generation of these cell types, calbindin expressing periglomerular neurons. So the next question came up when we went to the literature, shows actually quite a few correlations between VAX1 and PAX6. The Grus lab, the Stoikova lab, the Bertuzzi lab, they all showed that if you knock down one of these factors, the other factor extends its expression field. It has never been shown at the cellular, at the high resolution level, but in the forebrain, you see that you knock out VAX1, PAX6 invades the ventral brain, and vice versa. So in our system, we see the same thing. VAX1 shows a ventral dorsal gradient, PAX6 shows a dorsal ventral gradient. And we asked the question, is there actually a regulatory interaction between these two transcription factors? Does VAX1 regulate PAX6? Data is shown here. So here we use a VAX1 gain of function vector together with a co-attributed with a GFP expression vector to identify the transfected cells, transfected at P0 and analyzed at 2 time points, 2 days post-electroporation and 15 and 20, actually 3 time points, 15 and 25 days post-electroporation in the olfactory bulb after lateral and dorsal electroporation. Okay, lateral electroporation leads to a downregulation of PAX6 expression, mean intensity in cells in the subventricular zone, both in the lateral and in the dorsal aspect. And 15 days later, this lack of PAX6 expression is maintained we find many less PAX6 positive cells in the periglomerular layer of transfected animals when we use the VAX1 conditional allele. So PAX6 protein expression is reduced in the presence of VAX1. So of course, PAX6 is an inducer of dopaminergic fate. Is VAX1 now a repressor via its activity to repress PAX1 of dopaminergic fate? Basically, it's the same experiment. Overexpression of VAX1 combined with GFP expression and at P0 and an analysis 15 days later. When we do this in the dorsal aspect, we see that TH neurons generated here are strikingly downregulated. The second major population, which is generated here, calretinin expressing periglomerular neurons, are not affected by this expression. On the other hand, when we look into the lateral aspect, TH neurons are again strikingly underrepresented, while calbindin cells, which are dependent on VAX1 expression, do not show a change when we overexpress VAX1, showing that more doesn't make more. Okay, so this shows that VAX1 is a negative regulator of PAX6 and of course of dopaminergic fate. And we don't know if it's direct or indirect because of course, as I've shown you before, miR7 is doing the same job as VAX1. So leading to the question, is maybe VAX1 a positive regulator of miR7? And is the function of the negative regulatory function of VAX1 on PAX6 regulated or mediated by miR7? We don't have a clear answer to this question at this time point, but we have two indications. One is when we overexpress VAX1 in vivo in the lateral stem cell pool and look 2 days later, so we see a massive increase in VAX1, obviously, because we express it under the active promoter, but we see also a striking increase in miR7 microRNA, suggesting, not demonstrating, that there is a link between the expression factors. And the second argument we have is that there are actually in all, so miR7 is generated from 3 different genomic loci. And in the proximity of all of these loci, we find VAX1 binding sites, indicating, not demonstrating again, that there might be a link between VAX1 expression and overexpression or expression of miR7. Okay, so this is close to to the end of my talk. So I showed you how we use this good, well accessible system to study at very high resolution how different types of neurons are generated and how different transcription factors, probably via the regulation of microRNAs, control expression of this transcription factor and by this neuronal phenotype. We have other examples, for example, showing that Zik proteins are decisive factors between the fate of calretinin expressing GABAergic periglomerular neurons and dopaminergic periglomerular neurons. But for reasons of time, I will not go into detail concerning this work. So that's it. I hope I convinced you that this postnatal neurogenic system is a very convenient system to study different aspects at extremely high resolution, from proliferation, migration, differentiation to integration and function. And we will keep using it in the future. And if you want to collaborate to test your favorite neurogenic molecule in the system, this might be one way to look at this. Come to our lab and try to see and then try to use our system. We will teach you. That's all I want to say. So most of the work I presented today has been done by Nathalie Corré, who worked with Antoine de Chevigny to identify the PAX6 miR7 interaction, and Christophe Berclin, who was instrumental for basically all our work on microRNAs and also the gene expression screens that I presented. Marie-Catherine Tiveron worked on the Zik1, Zik2 project. Other members are implicated in other projects of the team. Key collaborators are Annalisa Ficco and Gunther Meister. Gunther Meister from Regensburg. We have a long-standing collaboration with Miltenyi Biotech in Cologne. Patricia Mellon provided the VAX1 conditional mutant mice. And we have a variety of very interesting and fun collaborations within the IBDM with Cédric Morange, Vincent Patron, and Pascal Durbeck. And so I don't show a picture of our building, I show a picture out of our building. So this is actually, well, it's a lie. It's a 10 minutes walk to go to the place where you have this view to the famous Calanques, the Parc National de Calanques. So we are actually located in the Parc National in Limini. And it might be a good place for a postdoc or a PhD for some of you listening to this talk. Thank you for your attention, and I'm ready to take questions.
Thank you, Eberhard. We have time for a few questions. I was wondering, Eberhard, whether you were able to follow the cells in the long term to know whether they integrate the olfactory bulb when you increase the number of TH cells.
We looked up to 60 days. So generally the phenotypes we observe, we look up to 2 months of age. Okay. And generally they are stable, so not coming back.
Thank you. Giuseppe?
Yes, thank you for this excellent presentation and this nice in vivo experimental system. You mentioned data about the interaction between VAX1 and PAX6 during eye development, all data from Greg Lemke and Stefano Bertuzzi. Yeah. I think at the time it was suggested that VAX genes could directly bind the PAX6 promoter and repress its transcription. So do you think that there might be also this kind of mechanism?
Yes, I'm actually
system, besides the microRNA mediated repression? Thank you.
Yes, actually, I'm sure that it's the case. So that I think the data is very clear that VAX1 can directly bind to the PAX6 promoter and repress expression. So VAX1 is, CMR7 is definitely one, it's what microRNAs do. MicroRNAs do never all or nothing. They reinforce a function, they fine-tune a defined function. So I'm pretty convinced, maybe I wasn't a bit clear about that. So there's no doubt that in other systems, in the ventral forebrain, in eye development, etc., there is direct binding of VAX1 to the PAX6 promoter. I also believe this is the case here. I believe that miR7 is a reinforcer, a fine-tuner of this aspect, which is what microRNAs do.
Thank you.
Thank you. Are there any other questions? In the audience? So we are going to close this session, and before I would like to warmly thank the speakers and the audience. So now there will be a lunch break.
Yes, Lydia, thank you. So the lunch break now, enjoy it. And we will come back for the next session at 2 this afternoon.